All right, let's dive right into this explainer. Imagine your average microscopic cell as a bustling, thriving metropolis. Now, any major city desperately needs a highly efficient way to process, manage, and ship materials, just to keep everything running smoothly, right? I mean, without a dedicated logistics hub, the entire system would literally collapse into total chaos.
And that brings us to a really fascinating logistical puzzle. How exactly does a microscopic cell ship its packages? We know for a fact that cells produce vital proteins and lipids, but how do these essential products actually navigate around inside something so incredibly tiny and yet so unbelievably complex? There has to be some sort of master mechanism for sorting and delivery.
So let's figure that out in Section 1, the cellular post office.
Meet the Golgi apparatus. This is the primary packaging and shipping center of the cell. If you look closely at its unique structure, you'll see it's composed of these distinct, flattened, sac-like layers all stacked together. Honestly, it kind of looks like a microscopic stack of deflated balloons, or maybe pancakes, but its function is absolutely critical to keeping the cell alive.
This little organelle operates exactly like a microscopic post office for the cell. It's the central hub where raw materials actually arrive to be processed. The Golgi takes these raw goods, modifies them, sorts them out, and then packages them up so they're perfectly prepped for whatever their final destination happens to be.
To handle all these cellular goods, the Golgi follows a really precise, highly structured three-step assembly line. Step 1, it modifies the incoming proteins and lipids. Step 2, it meticulously sorts these materials based on where they need to go. And finally, Step 3, it packages them into these tiny structures called vesicles. You can basically think of these vesicles as little delivery trucks used for transport within the cell, for secreting things outside, or even to build other really important cellular components, like lysosomes.
Let's step back a bit for Section 2, mapping the cell city. If we map out different cellular neighborhoods, say a bacterial cell, a plant cell, and an animal cell, we can see exactly where these structures actually sit. In complex plant and animal cells, the Golgi body is a huge, prominent landmark, nestled right in there alongside the nucleus and the endoplasmic reticulum. But if you look at the simpler bacterial cell, its internal landscape is quite a bit different.
Comparing these side by side reveals a major distinction in how cellular infrastructure works. While complex plant and animal cells totally rely on this dedicated shipping center, the Golgi apparatus, much simpler bacterial cells actually do not. Bacteria just completely lack that same level of specialized internal compartmentalization. Now, the crucial takeaway here is that the Golgi doesn't operate in a vacuum. It's a highly connected transit hub. It functionally links right to the endoplasmic reticulum, which is the factory where a lot of these proteins and lipids are first manufactured. It also connects directly to the cell membrane for when packages need to be shipped completely out of the cell, which ensures the whole cellular city stays supplied and perfectly synchronized. Which brings us to a wild piece of history in Section 3, the doubted discovery. I want you to meet Camillo Golgi. He was a pioneering Italian scientist and the very first person to spot this mysterious thread-like network. He made this discovery way back in 1889, using some highly innovative staining techniques, while he was actually examining the nerve cells of a barn owl. Just imagine the sheer patience required to peer through a late 19th century microscope and identify a complex microscopic network that literally no human being had ever documented before. But you know, the path to scientific truth is rarely simple. After Golgi first observed this structure, the scientific community pushed back hard. During those early years, the very existence of the apparatus was heavily doubted. Why? Because the light microscopes of the time simply could not resolve the structure clearly enough for anyone else to verify it. It wasn't until decades later that his initial findings were conclusively confirmed, and that was entirely thanks to the invention of powerful electron microscopes. Talk about ultimate vindication, right? His eventual triumph cemented his legacy in cellular biology forever. When the structure was finally undeniably confirmed by the broader scientific community, it was officially named the Golgi apparatus, right in his honor. It's just a perfectly fitting tribute to a scientist who completely trusted his own observations, even when the technology of his day couldn't back him up. So I want to leave you with this final thought to ponder. If it took decades of technological advancement just to prove to the world that the Golgi apparatus actually existed, what else might be hiding right now? What other invisible structures, mechanisms, or crazy cellular secrets are simply waiting for better technology to be discovered? sitting just beyond the reach of our current microscopes. Keep that curiosity alive, and thank you so much for joining me for this explainer.